Hello everyone, welcome back to Epidemiology and Biostatistics. So today's topic is types of epidemiological studies and I am Dr. Muhammad Razif Shahriar. So by end of this lecture, you should be able to describe basic study designs used in epidemiology studies, explain the advantages and disadvantages of each study design, and finally differentiate each study design. Study designs direct how the investigation is conducted. The main common goal of epidemiology studies is understanding the frequency, pattern, and causes of disease in the population. Therefore, it has high reliance on the survey method and rooted in the concept of the population. The underlying or base population is the starting point in a study design. There are five major types of study design. The first one is case series, mostly clinical and also in population. The second study design is cross-sectional, case control study, cohort, you have prospective and retrospective, and finally you have trial. This slide explains why do we need different study design. So if you look at descriptive studies, it is mostly to develop hypotheses. And then when you move to case control studies, it investigates its relationship to the outcome. Cohort studies define its meaning with the exposure, and finally, clinical trial tests the link experimentally. Therefore, there is an increasing knowledge of disease or exposure when you move from descriptive studies to clinical studies. This is another slide that summarizes the relationship between study design and time. In cross-sectional study, the time frame that we are interested is the present time. For prospective cohort studies, it is moving from the present time to future, means that we follow up the population. In retrospective cohort studies, we go back in history to the past, from present time to the past. Similarly, case control study also is actually a retrospective study which are interested in what happens in the past. Whereas in randomized control trial, we have to treat the patients and give placebo to controls, and we follow up to the future. In earlier series of lecture, we have learned that epidemiology studies are divided into two types. The first one is descriptive and second one is analytical epidemiology studies. So let's look at the study design involved in descriptive epidemiology studies. There are three types of descriptive epidemiological study design. The first one is case study, second one is case series, and the third one is cross-sectional studies. Let's see what is case study. Case study is a detailed presentation of a single case or a handful of cases. It generally reports a new or unique finding, example on previous described disease, unexpected link between diseases, new therapeutic effects, adverse events. The case may be an individual or an event, a policy, etc. There are several research values of case report. The first one is to illustrate or support a hypothesis. Example is atlas subluxation. Atlas subluxation is a minor misalignment of the atlas, the very first bone on our neck, C1, in a man with gastritis. So when we describe this atlas subluxation in a man with gastritis, we are illustrating or supporting a hypothesis in a case report. The second value is to prompt a new hypothesis. I'm going to use the same example. Atlas subluxation can cause gastritis. So when we do this kind of research, we are prompting a new hypothesis. The third one is report treatment failures. Example, correcting atlas, the C1 bone in the neck, did not relieve gastritis. And the fourth one is to report iatrogenic reactions. Example, gastritis became better when we correct for atlas subluxation, but the patient developed brain tumor. So all these can be reported in a case report. I would like to share with you this figure which shows the hierarchy of a research design. So we can see that case report is the largest scheme of things and it is at the bottom of the hierarchy of research design. The highest hierarchy is randomized controlled trial and meta-analysis. So why case studies and reports are low on the totem pole? First one is it's only a preliminary observation which are frequently later refuted. The second reason is it may rationalize questionable treatment. Example, whether glycemic index diet versus carbohydrate counting among gestational diabetes is effective or not. The third one is there is a bias reporting in case studies and case reports where negative studies may not be published. And the fourth reason is it's not experimental because it is just a descriptive epidemiology study. And finally, it does not have a control arm. So this slide shows examples of case reports classified according to their use. The second study design under descriptive epidemiology is case series study design. Case series study design shows the experience of a group of patients with a similar diagnosis. In case series, it assesses prevalent disease, and cases may be identified from a single or multiple sources. Case series generally report on new or unique condition, and case series study design is a realistic design for rare disorders. The advantages of case series studies is it is useful for hypothesis generation, it's informative for a very rare disease with few established risk factors, and the third advantages is it characterizes the averages for disorders. The disadvantages of case series are it cannot study cause and effect relationships, it cannot assess disease frequency, and finally, it does not allow comparison with those without the condition or treatment. Let's move to cross-sectional study design. Cross-sectional study design is a type of observational or descriptive studies where the researcher has no control over the exposure of interest, example, the diet. Cross-sectional studies involve identifying a defined population at a particular time and measuring a range of variables on an individual basis. It can include past or current dietary intake, and data may be explored in relation to the presence or absence of disease or health outcomes. Cross-sectional study design is not based on hypotheses. Therefore, it is not possible to determine whether the exposure and the outcome are causally related because the data represent snapshot of information at one point of time. In cross-sectional studies, an observational design surveys exposure and disease status at a single point in time. It's a cross-section of the population. So the study only exists at this point of time in the blue bar, and the researcher has no control over the exposure of interest, example diet, in the future or in the past. So in a cross-sectional study, we will approach the study participant and identify those with disease and without the disease. And among those with and without the disease, we will see whether the factor is present or absent. So, because the study only exists at one point of time, it's not possible to determine whether the exposure and the outcome are causally related. and data represents only the snapshot of information at one point in time. So why do we choose cross-sectional study design when the purpose of the study is descriptive and there is no hypothesis? Example, when you are interested to do a survey, and the purpose is to find the prevalence of the outcome of interest for the population or subgroups within the population at a given time point. Cross-sectional study is also known as prevalence study because it can be used to estimate prevalence of disease in a population. So, if we remember back, prevalence is number of cases in the population at a particular point in time, expressed as a rate. So, prevalence is equal to number of individuals having the disease at a specific time divided by number of individuals in the population at the point in time. So what we are looking at the screen is an example of a cross-sectional study. The title of this paper is Nutritional Status and Dietary Intakes of Children aged 6 months to 12 years, findings from the Nutrition Survey of Malaysian Children, CNAT Malaysia, which is published in British Journal of Nutrition in 2013. So this is a cross-sectional study because it studies the Malaysian children where 3,542 children were involved in this study. And the finding shows the prevalence of overweight is 9.8% and obesity is 11.8%, which was higher than those of thinness 5.4% and stunting at 8.4%. So this kind of study gives us prevalence. Due to its popular use in research studies, cross-sectional studies have several uses. The first one is Prevalence Survey. It is commonly used to describe the burden of disease in the community and its distribution. The second use is to describe population characteristics. It is commonly used to describe population characteristics, often in terms of person, who, and the place, where. Example shown in this slide is on a research on food consumption pattern findings from Malaysian Adult Nutrition Survey. So this study describes the food consumption patterns among Malaysians. The third use is in migrant studies. So it gives clues as to the association between genetics background and environmental exposures on the risk of disease. Example here is fat storage syndrome in Pacific peoples, a combination of environment and genetics. Cross-sectional designs can also be used to study knowledge attitude practice studies, where it is a purely descriptive and help to build up a better understanding on the behavior of the population. KAP studies is conducted without necessarily relating to this to any disease or health outcomes. Example here is a study published on diet, nutritional knowledge, and health status of urban middle-aged Malaysian women. The final use of cross-sectional study is developing hypotheses. Cross-sectional studies is used in developing hypotheses when the work has added support to other findings. And also, it is used in developing hypotheses when the results have encourages further work in the area studied. Nevertheless, cross-sectional studies indicate association that may exist and are therefore useful in generating hypotheses for future research. The example in this slide is a paper published on intake of dietary plant sterols is inversely related to serum cholesterol concentration in men and women in an epic Norfolk population, a cross-sectional studies. So in this study, a hypothesis is developed and tested whether there is an inverse association between plant sterol and serum cholesterol among a population in epic Norfolk. It seems that there are several advantages of cross-sectional studies. The first one is it is relatively inexpensive and takes up little time to conduct. The second advantage is the prevalence of outcome estimation can be done in cross-sectional studies. Many outcome sources and the risk factor also can be assessed in cross-sectional studies, and cross-sectional studies are useful for public health planning, understanding disease etiology, and for the generation of hypotheses. Finally, in cross-sectional studies, there is no loss to follow up because it only studies at a single point of time. However, there are several limitations of cross-sectional studies. The first one is the exposure and the disease or outcome are measured at the same time, where it is not possible to identify which is the cause and which is the effect. Therefore, to test a hypothesis, a cohort study will be needed. The second limitation of cross-sectional study is confounding factors may not be equally distributed between groups being compared, which may lead to bias and subsequent misinterpretation. So what is confounding factor? Confounding factor is a variable which is related both to the exposure, example the diet, and the outcome of the disease. Examples of confounding factor are age, gender, and ethnicity. The impact of confounding factor is removed by stratifying the sample by confounders at the analysis stage. However, the residual confounding related to variables poorly measured or not at all measured is still possible. Other limitations of cross-sectional studies are current diet may be altered by the presence of disease, where the outcome or disease is having influence on the exposure diet variable. Diet also may change over time when the subjects are not affected by disease. Limitation of cross-sectional studies can also happen due to error in recall of the exposure and possibly the outcome. And finally, prevalence studies may be affected by Neyman bias, which in Neyman bias, subjects are not included in the prevalence measured because they have died early from the disease or the symptoms have gone.